Today's mini conference will be on honeybee pathogens, so viruses and Chalkbrood. We'll start off with Dr. Marley Iredale. So she's a veterinarian pathologist working on expanding her knowledge of honeybees and their pathogen. She received her bachelor degree in wildlife ecology in veterinary and a veterinary degree from Washington State University. She completed a one-year medical internship at a private veterinary hospital in New York before her residency in Anatomic Pathology at the University of Florida, where she fell in love with honeybees as part of her entomology department. Her Ph.D. work aims to mitigate some of the challenges currently facing honeybees and the apiarists. So you should be able to share your screen. For the attendees, if you have questions, please use the Q&A, okay? And then after Marley's talk, what we'll do is we'll have a short Q&A.
Thank you so much, Etienne. I appreciate that kind introduction. And thank you so much for sharing the link. My dad is here, so hi, Dad. But yes, like Etienne said, I am a veterinarian, and I have done specialty training in anatomic pathology. And mostly what that means is I specialize in epidemiology and disease dynamics and things like that. But I discovered honeybees kind of halfway through my residency and decided that this was the niche that I wanted to fill. And in the United States right now, we're kind of facing a crisis because they changed a lot of our legislation to do with antibiotic use for honeybees. Now you have to have a prescription from a veterinarian to be able to apply antibiotics to your hives in the US. But the truth is, veterinarians know nothing about honeybees. We're really good at comparative medicine, but you really do have to have some fundamental background in the system to really be effective as a doctor for honeybees. And so that's why I've come back to do my PhD, is kind of fill my knowledge gaps and to make some connections with apiarists and other people in the field. I'm going to be talking about viral diseases of honeybees, and this is the focus of my dissertation. I work primarily with Israeli acute paralysis virus, but I do a little bit with deformed wing and black queen cell virus, so I'll talk a little bit about those as we go. I know we probably are all a little bit more familiar with viruses than any of us really wanted to be or expected to be after the past 2.5 years or so. But there are some important points about viruses that I just would like to reiterate for the sake of the conversation that we'll have this evening. Don't forget that viruses are essentially the least complex replicative organism, if you can even call them that. They're just strands of nucleic acid, oftentimes wrapped in some coating of protein. And these are able to infect host cells, and then they often utilize actually the host machinery to replicate themselves. So they're really just a blueprint. But one of the problems with this is when they hijack host machinery, a lot of times that takes away from host functions, and it incites an immune response, and that's why we see the clinical disease that we're all pretty familiar with. As far as honeybees are concerned, there are at least 20 that have been identified, but I will be completely honest and say that we know very little about those viruses. There's probably only, I would say, 6 to 8 that we really have some good data on. So for me, when I'm approaching honeybees, when I'm approaching honeybee medicine, I'm always kind of thinking from a diagnostic standpoint. And for me, one of the most important steps in viral diseases is transmission from one host to another, right? They can't live outside the host. They need a host to replicate and perpetuate themselves. So transmission from host to host is incredibly important. And then again, even when looking at this flow diagram, we're thinking about where can we as apiarists intervene to help to try to prevent some of these transmissions. So for me, the big thing, the thing that I will say over and over again is good biosecurity between apiaries. You know, cleaning equipment, making sure that solid queens are going out to other apiarists, things like that are so important. I think that we have all recognized how horribly destructive varroa mites have been, and a lot of the spread of varroa is probably due to human movement of honeybee colonies. So again, good biosecurity is so incredibly important. Currently, one of the main ways in which we try to disrupt some of these viral transmissions is to replace the queen bee. There is a lot of evidence that she does pass viral infections to eggs. So replacing her can sometimes be a way to mitigate severe infections in a colony. Drones, they're not worth very much, but they are a way in which viruses can be transmitted. And so a lot of groups are currently working on doing artificial insemination of queen bees. And what that allows us is one drone. So you can really limit the source and all of the different types of pathogens that could go along with that seminal fluid if you limit it to one drone. And finally, of course, the big baddie, varroa. We're all very familiar with this. Keeping our varroa under control, trying to mitigate some of the side effects of severe varroa burdens is really, really huge in helping prevent really bad viral diseases. So this is just kind of another way of looking at that infographic. If we think about who our most susceptible cast of bees in a hive is, it's probably our worker bees, right? They're involved in absolutely every aspect of the colony. And unfortunately, these girls are exposed to viruses probably from the minute that they are oviposited. So like I said, we think that the queen bee is able to transmit viruses vertically to her eggs. Worker bees are going to be coming in and feeding those new larvae. That's another source of possible viral infection. We see varroa again. These are going to infect the cells in which the bees are pupating, and then go out with them and be transmitted all over the place. These girls clean their own cells when they emerge, so that's a good way to interact with even more pathogens. And then they work in the colony for a little while. They're undertaking, so they're encountering pathogens when they're removing dead bees from the hive. And then of course our foragers are really susceptible, and they do drift. So bees from an infected colony drifting into an uninfected colony can perpetuate these diseases. This is a pretty busy infographic, but I put it up here to just continue to reiterate the importance of varroa. So what this graph is showing, that we are pretty sure that varroa started in Asia in the early 1900s and then has kind of swept across the globe with managed honeybees. And from the genetic analyses that we've done on deformed wing virus, we know that it followed a very similar pattern. So you could argue chicken or the egg in this case, but I think based on the biology, it is highly suggestive that deformed wing virus followed varroa around. Again, this is a little bit of a busy infographic, and I mostly am putting it up here because I am going to start with an apology and say we really don't know a ton about viral diseases in honeybees. It's a really big avenue of investigation for a lot of different groups. So my genuine hope is that we're going to start to, fill in all of the question marks in this graph. But the truth is we don't know a lot, and it's a really complex system to try to tease apart. So generally when I give this talk, I'm talking to a room full of people and it goes a little bit more smoothly. But this slide is my opportunity to reach out to you guys and ask what signs of viral infection you've seen in your colonies. And please, my email address is available. Please reach out to me if you've had severe viral disease. I'd love to hear about your experiences. But these are kind of like the 6 general signs of viral disease. They're very non-specific, and you'll notice I'm going to keep repeating these signs for multiple different viruses. But kind of in general, things that we think about are definitely darkening of the abdomen and hair loss. Those seem to be, for whatever reason, common to all viral diseases. But weakness, trembling, and inability to fly can be associated with several different diseases, but those are kind of things that we lump into what we call the paralytic viruses, which is somewhat misleading of a term, but as far as we know, those viruses cause weakness that then leads to the bees not being able to perform their normal roles in the colony. I think that this is a really important point to make. I know colony collapse disorder hasn't really been a thing that we've talked about a lot lately, but I still do get questions about it, and so I like to include this slide. Colony collapse disorder was really a big deal probably at least 10 years ago now, and essentially what people noticed was just massive egress of bees from their colonies. Essentially everybody would pick up and leave except for maybe a couple of workers and then whatever brood was left behind. And we don't know where they went. We don't know why they left, and they didn't come back. But we really still don't know what caused this kind of mass exodus. And we haven't really seen it again. So it makes it even more challenging to try to figure out what happened. But in cases of viral disease, you will generally see signs of infection. So those things I listed on the previous slide, as well as mortality in the brood. But you also will typically see dead bees. So they're going to be dying in the hive or around the hive, and then our undertaker bees will be removing them, just like with any other pathogen. So colony collapse disorder and that investigation has kind of led us down a different path. Instead of it just being one syndrome, it really does seem to be multiple different threats facing honeybees all at the same time. So increased use of commercial pesticides, climate change, introduction of new pests and pathogens, and then ecosystem destruction are all really challenging for honeybees. And I'll put in a plug for native bees. It's really bad for native bees too. But for me, specifically, I look at the diseases of honeybees, and that's kind of what we're going to focus on, but do be aware that all of this is multifactorial. So if you have a lot of environmental stressors, your bees are going to be a lot more susceptible to disease. If there's a really high pesticide burden in your area, and so on and so forth. So as far as knowing what kinds of diseases are in your area, I just want to put in a really big plug for Bee Informed Partnership. I'm not sure if they have a Canadian branch, but here in the US, this is the organization that does the most monitoring. So they have presence in pretty much every state. They look at almost every pathogen that we know of or that we have a diagnostic test for. And they don't have a ton of buy-in from apiarists. So sometimes their sample size is a little bit low, but they do have a Sentinel apiary program. So if you're in the US, you can sign up to help with that. And I really do believe that citizen science is going to be what helps us solve a lot of these problems. There's just too many colonies, there's too many bees for researchers to manage alone. Buy-in from beekeepers is huge for us. Now I'm going to go into kind of my discussion of the individual viral diseases. Like I said, we don't know a lot, and it will be a little bit redundant because a lot of the clinical signs are very similar, but I do want to at least draw everybody's attention to some of these really common viral diseases. So this is a virus that we have here in Florida. This is actually a picture from our research apiary that my technician took, I think 2 weeks ago now. But so this is a viral disease specifically of queen bees. Again, we don't really know why, but for whatever reason, it infects the queen larvae and it turns them this really dark black color. And it can actually also change the color of the wax to be black as well. That's the name of the virus. It does kill the queens. And so this is pretty devastating if it gets in queen rearing operations. My understanding is it's a little bit more common in Australia, but we do have it here in the US as well. Chalkbrood virus was probably the first honeybee virus that was really well characterized. And I think that that mostly stems from the fact that it really closely resembles the bacterial diseases that we worry so much about. So as you can see, it causes, similar kind of melting of the larvae or the prepupae. We think that it's mostly so Chalkbrood virus. So we think that it's transmitted in brood food, which makes sense if you consider the fact that it mostly affects larvae. But generally speaking, this is an asymptomatic infection. So this, although this is primarily a disease that affects larvae, it does cause shortening of the lifespan of the adult worker bees, which can have a negative impact on the colony overall. And again, the larvae tend to look like what you would expect from a virus called Chalkbrood virus. They either kind of melt into those little gooey puddles or they dry out and become this kind of husk, dark brown husk. Deformed wing virus, we talk about it here in the honeybee lab as being like one of the most significant threats to honeybee health, but I think that this is a conversation that we all kind of need to have as a group. Almost every colony that you check will have some level of deformed wing virus. It's endemic now. And yes, a lot of that goes along with Varroa, but Varroa is endemic now too. And so I think that this is going to end up being something that we just deal with going forward. Really good mite control, keeping really strong colonies, things like that will prevent deformed wing virus from being a really big problem in your colonies. But it is around. You will see, you know, deformed bees with deformed wings in your colonies and things. And I try to tell people, you know, don't freak out, but it means that there's probably something going on that you should address, you know, either improve your nutrition, or I'll say it again and again, I'm a broken record, treat your varroa. This is classic, right? This is the disease that everybody talks about because it's got such beautiful clinical signs. The bees emerge with deformed wings. The other parts of their anatomy can sometimes be misshapen. They're usually lacking in hair. These newly emerged bees should be really fuzzy. It does cause increased mortality in the worker caste. So we do think that it has some effect on the colony as a whole. And then of course, any of these girls with deformed wings are not going to be super effective in the colony. They can continue to do things like care for brood and, you know, receive resources from foragers and things, but they really aren't going to have the full productive life that they should. Varroa destructor virus 1 is really closely related to deformed wing virus. And if you're reading some of the old literature, this will probably be included as a subspecies of deformed wing virus, but it has been reclassified into its own group. But of course, since it's newly reclassified, we don't really know a lot about it. However, it is rapidly spreading in North America, and we are concerned about what it's going to do when it is introduced into US colonies. So this is well, I work specifically with Israeli acute paralysis virus, but everybody kind of lumps Israeli acute paralysis virus, cashmere bee virus, and acute bee paralysis virus together. And the reason for that is when they were originally characterized, everybody thought they were the same virus, right? The availability of genetic testing now has kind of allowed us to put all of these things into different boxes. But the truth is, at the end of the day, those boxes are human constructs. So we talk about these viruses together because they cause really, really similar clinical signs, like we talked about in that initial discussion: trembling, paralysis, weakness, the paralytic viruses. The only one that I am a little bit unsure as to why it's included in this group is cashmere bee virus. We know next to nothing about this virus, but it actually causes them to grow like really long, shaggy, furry coats, which is really unusual for a viral disease. So again, this is kind of what you can expect, you know, really heavily infected viruses or bees to look like. They lose all of their hair, and they do get pretty significant darkening or melanization of the thorax and the abdomen. Again, to bring us back to colony collapse disorder briefly, when it was first described, Israeli acute paralysis virus was the pathogen that was most often identified in infected colonies. But again, that's never been recapitulated. It was a really weak association at the time. So we really don't talk about IAPV in the context of colony collapse anymore. Chronic bee paralysis virus is a really interesting virus. And again, we don't know a ton about it, but for some reason, it seems to cause two distinct disease syndromes, which again is very untypical for a viral disease in honeybees. They're relatively similar, but have a little bit of a difference in clinical signs. And we don't really know what that is. We don't know why, you know, one colony experiences one syndrome and another a different one. But all of these are associated with chronic bee paralysis virus. And this is actually the only virus that I know of that has caused collapse of a colony in a person that I have actually spoken to. So I think that this is prevalent, and I think that this is important, but we just don't know a lot about it yet. This is another relatively new introduction to North America, Lake Sinai virus. There is a 1 and a 2, but 2 is the one that we have in the US right now. Again, it was only recently introduced, so we don't know a lot about it, but it is spreading, and they have associated it with colony losses, although the connection to colony collapse is tenuous. So finally, I just want to kind of put this last list of viruses up here. They only have one line because that's pretty much all we know about them, is that one line. But if you're reading the literature, if you're reading textbooks, these are going to be viruses that you're going to come across. And then other than what I have listed here, their significance as far as apiculture as a whole is not really very well known. Honeybee immunity is something that I could talk about for an hour in and of itself, but I just want to put it up here because I think it's so cool. And I think it's really amazing how they're able to kind of combat some of these diseases in ways that wouldn't even occur to a vertebrate. So a lot of their immunity has some analogy to what we have as vertebrates. They have a cuticle, which is similar to our skin. Their gut morphology is very similar to ours. So that is a barrier which ingested pathogens are kind of blocked from entering circulation. Instead of white blood cells, they have something called hemocytes, but they serve a very similar function. And then just like us, they have antimicrobial peptides and enzymes that can help them fight some of these infections. But what I think is really, really cool about honeybees is their social immunity. So if you think about the colony as a whole, how it prevents infection or how it reduces infection. So one of the very first things is the basic aspect of honeybee biology, that their castes are separated by space and time. So if you think about the most high-risk individuals, your foraging bees, they don't spend a ton of time in the hive. They come back to the entrance, they pass their resources off to receiver bees, and then those bees actually go walk on the frames, put things in cells, interact with brood and stuff like that. So your brood and your newly emerged workers, who are theoretically, the weakest and less able to survive these viral diseases, are kept as far away from your foragers as possible. They also groom each other. So I think there's been a lot of emphasis put on strains of bees that are able to remove varroa. So those kinds of hygienic behaviors, grooming one another, grooming themselves, are really a good way to help keep things from entering the colony. There's another interesting aspect of honeybee immunity called altruistic self-sacrifice, which is an acronym we shouldn't write down. I've called it self-removal up here, but it's this innate instinct in sick bees to take themselves away from the hive. So worker honeybees that are infected, that recognize that they're infected, leave and they don't come back. And this is kind of similar to our undertaker bees, who are removing infected brood or dead nestmates and keeping the colony nice and clean. And all of these things are so effective. Honeybees can prevent a lot of different diseases from taking over their colonies just with these basic aspects of their biology. And with our help, they do even better. So as far as what you do if you think that you have a viral disease in your colonies, here in the US, our best resource is this bee research laboratory in Beltsville, Maryland. They're a really great group. From what I understand, they will receive samples for free, and they'll give you, kind of a full breakdown of whatever they find in your colony. In Florida, we've just hired a new research technician, and his whole position is going to be to develop a diagnostic lab here at UF. So we're hoping that that's going to be another place where people can send bees for diagnostics. And then in truth, the deep wish of my heart is that veterinarians are going to be a good resource to people in the future once we get everybody cut up to speed on how to take care of honeybees. Our background is in epidemiology and in disease investigation. So I think that this is a place where we can really help, and that we've been underutilized so far. As far as what you can do if you think that you have a viral infection in your colonies, the thing that I can say definitively is keeping your colonies strong is the best way to prevent disease. So supplemental feeding, requeening colonies, controlling varroa, hygiene between apiaries, these are all, pretty fundamental things that I think that we all can do, but that can have a really, really big impact. If this is something that you want to learn more about, please reach out to me. I'm more than happy to talk about this anytime. It's my passion, it's my project. But as far as independent learning, I think that this is probably one of the best books that we have available right now as far as disease ecology goes. They took a really academic approach. I'll say it's very thorough, but don't try to read it before bed. You will fall asleep. But it's really great. And I think it's got, all of the information that we do have all in one place. This is actually the book where that big table of viral diseases came from. So I really do think it's a great resource. As far as contributing to honeybee virus research, like I said, I think citizen science is the greatest way that any of us can contribute. And so I really do encourage everybody to, go and check out Be Informed and consider signing up for their Sentinel Apiary program if it's available in your area. And then again, you can always reach out to us at UF, and we're always happy to talk. I'd now be happy to field any questions.
Thank you very much for that. It was good, very good.
Thank you.
So the floor is open to questions. Ian, go ahead.
Yeah, I'd like to ask you maybe just to speak a little bit on, you covered it in the immunity slide there and the way bees control a viral infection. There's discussion a little while ago in regards to hygienic behavior and how bees would cannibalize the brood and infect the rest of the bees within the colony, and everybody got an uproar. Oh, we can't be breeding our bees for hygienic behavior because we're doing ourselves a disservice. But in actual fact, on your slide, you mentioned a few points there and just the mechanisms the bees use to be able to manage that infection, like that infection issue. Could you speak a little bit about that?
It is a very complicated issue. And I think that hygienic behavior is one of those things that has different definitions to different people. And so just to define the term how I understand it, my understanding of it is always in the context of varroa. We talk about hygienic behavior as, their ability to remove varroa. If it's also discussed in the context of, the idea that they're removing dead and infected brood, I think that adds a whole nother element. I can't say definitively, but I think that cannibalization and removal and exposure in the process of removal, unfortunately, are just always going to be part of it. And there have been some attempts to try to model this system mathematically or with statistics. And the truth is, reducing transmission is the only way to control these diseases. And unfortunately, I don't think that transmission in the process of cleaning up the colony is going to be a place where we can intervene. Does that answer your question?
Yeah, and I also heard people talk about as the bees are cleaning out the nest, they almost segregate themselves. I think you touched on it as you spoke. They kind of like they clean out the infected brood to dispose of it, and they kind of isolate themselves away from the rest of the bees, or even dispose of themselves. What do you think about that?
I think it's amazing. Yeah, so that's what we call altruistic self-sacrifice or altruistic self-removal. And it's a phenomenon that we see over and over. And again, I think that we need to do more to understand what the cue is for that and how they recognize that cue. And maybe that would be a good way that we could help to kind of use that information. But I think it's amazing. And we do see it in a lot of different pathogens.
Okay, thanks for that.
If there's time and you're inclined, Marley, would you mind sharing a little bit about your project specifically? I'm super interested.
That's so sweet of you to ask. Yes, I just happen to have a few slides at the end of my presentation, just in case somebody asked me.
If you want to take the time now, and folks can write some more questions, and if you want to do an update on your project first.
Thanks. Yeah, so the focus of my dissertation is Israeli acute paralysis virus. It's widespread in honeybee colonies. Again, I mentioned that it was implicated in colony collapse disorder, which we're not super convinced of anymore, but it certainly does contribute to overwinter colony losses. This is endemic, but it mostly causes subclinical infections. It's only when you get really high viral titers that you see those kind of classic clinical signs like paralysis and darkening and hair loss and death. But one of the challenges with this virus is we know next to nothing about it. There have been quite a few surveys that describe who in a colony has viral particles. So, we know it's present in drones, we know it's present in workers, we know it's present in queens, it's present in a lot of hive products, but we don't know anything about how it's transmitted. We don't know anything about what level of viral infection you have to start to see some of those clinical signs. Next to nothing, unfortunately. And so the goal of my project is to kind of try to expand those gaps that we don't know anything about. So it's a 3-part dissertation right now, and we're going to try to describe transmission. So really fundamentally find out, how does it get from one bee to another? And then we're going to do some microscopic investigations to kind of look at the tissues and see, okay, how does it cause paralysis? Is it actually infecting the nervous system? Is it, causing changes to the ion concentration in the hemolymph so they can't move their muscles? Why does that actual paralysis happen? And then we have a colleague who has been working on human viruses in the same family and who thinks that they may have found a compound that can inhibit replication of that group of viruses. And so they'd like for us to try it in honeybees to see if it would work, because that would be really good for their group. But kind of like how to approach this question of fundamental viral transmission. Essentially, think about every way that a honeybee could encounter virus. We know that it's passed via varroa mites, but how? Is it when the varroa mite feeds? Is it injected with the saliva? Is it something they're exposed to because the mites are passing it in feces, and as the bees clean their cells to emerge, they're picking it up? Is it only transmitted when bees participate in trophallaxis and feed each other with saliva? So like any kind of fomite that you could think of that a bee could encounter a virus in, we're going to test and see if they have viral particles. But kind of the second part to this that we need to think in mind is just because the virus is present doesn't mean it's transmissible. All of those things that we talked about honeybee immunity are ways to kill the virus. So even if virus is there, it's not always infective. So then you take all of these samples that you found to be positive for virus and you expose new bees to them. And if they start to exhibit clinical signs, then yes, that is truly a route of transmission. And one of the interesting things that I would really like to investigate more is this difference in summer and winter bees, which I would love to hear our Yukon colleagues comment on. But here in Florida, the actual physiology of the bees is different between summer and winter. And for whatever reason, winter bees seem to be a lot more resistant to things like viral disease. And we don't know why that is. We don't even know what's really different about them. They all look exactly the same, but they have a little bit different behavior. They have a little bit different physiology and metabolism and things. So there is some sort of difference there.
Marley?
Yeah.
You may be interested. Back during CCD, I was working with biologics from Israel. Mm-hmm. And I went out and collected bee samples from a colony that was collapsing from CCD. And you're welcome to email me for a perspective on living through CCD and what we actually found. Cool. And also on signs of viruses too. If you want to email me, I will. There's a number of things that you said that could stand some. Okay. Anyway, we collected bees with the virus. I cultured it in pupae by injecting the pupae, and then sent it back off to Israel. They purified that virus and sent me back the purified virus, and then had me do cage trials to do oral inoculation via sugar syrup to figure out how big a dose to give to colonies in order to infect them with it. And I'll tell you, it did not take much in cage trials in dilution. So they did not need to come in, via a mite. So oral transmission did work very well for that strain. And apparently, we suspect it was a particularly virulent strain of Israeli paralysis virus in combination with Nosema cerani infection. And CCD correlated extremely well with the invasion of Nosema cerani. And for your information, the only pathogen that causes the symptoms, that caused the symptoms of colony collapse disorder, where it dwindles down quickly to that handful of bees and queen, is Nosema. That's the only pathogen that's the distinctive symptom of advanced Nosema infection, that sudden dwindling. I was just looking at for Nosema this morning, or just an hour ago, actually, from coming back from the field. And then we inoculated a whole yard of bees with that strain of Israeli paralysis virus, and then put in a dsRNA inhibitor of it, and tested the different groups. And it was really obvious the difference between colonies. I could easily tell. We had I was blinded to treatment, just color coded, and we could very quickly tell by just the color coded different groups what they had gotten as far as treatments, and the importance of on how on colonies with Nosema ceranae, inoculating them with that Israeli acute paralysis virus. It was really the only in situ trial ever where CCD was actually created in situ under controlled conditions. And we collapsed a whole bunch of colonies with all the signs of the classic signs of CCD, which is very different than what they call CCD nowadays, which is the disappearance of the bees. Let me just say one more thing. With what I've noticed as a beekeeper and a researcher, there's very few viruses that actually you ever see dead bees within the hive. Okay, so chronic bee paralysis virus, that is very typical on not the hairless black symptom of it. There's two different types of chronic bee paralysis virus, but you will see dead bees and trembling bees from that one. But deforming virus, you very rarely see dead bees within the hive.
Yeah, I think you have to have a really severe burden of DWV and Varroa to actually collapse a colony that way.
Oh, I was showing one to my assistants today, a colony in the middle of collapse from Varroa and deforming virus. And the symptom, especially, you don't see it so much in the adult bees, you see it in the brood, in the pupae of deforming virus, and then likely other paralytic viruses also. But the sign with this parasitic mite syndrome is very easy to point out to them. We happened to come across a colony that was collapsing from a high Varroa load.
Oh. Thank you so much, Randy. I do really appreciate your insight, especially since you have so much experience, especially with that specific disease. Thank you.
Certainly.
This is what I do. This is my background in veterinary pathology, and so this is what a microscopic slide of a honeybee looks like. Specifically, the head of a honeybee. Every image on the left of the screen is a normal honeybee, and everything on the right was exposed to a really high level of pesticides. But I just use this as kind of an example of how this can be a diagnostic tool for honeybees. But so this image A is, a low magnification view of the head, and beautiful optic lobe going out to this beautiful compound eye. And what I'd like to draw your attention to is these kind of daisy-shaped structures. These are all of the hypopharyngeal glands. So what actually makes brood food. And the image C is a higher magnification and D is even higher. But these are an epithelial cell and they're making secretory products. So all of these little pearls are secretions, our enzymes are going to go out and become brood food. So these are really beautiful and healthy. They've got nice crisp borders. Everybody is where they're supposed to be, doing what they're supposed to be doing. And then this panel of images on the right is our bee that was exposed to a high dose of pesticides. And I apologize, it's not an exactly analogous image. We're a little bit off center. But at the higher magnification view, you can start to pick up these cells don't look the same. They're all really shrunken. They're not making any secretory product. Their nuclei are really smudged. Their edges are indistinct. So this is what, unhealthy hypopharyngeal gland looks like microscopically. Image F is even closer. And I bring this up just to mention that if you're reading the literature, you will find a lot of papers that are claiming that X or Y has a negative impact on honeybees because of changes to the hypopharyngeal glands. And I just would like it to be known, this is a really non-specific lesion. So we see changes to the hypopharyngeal glands in almost any stressor of honeybees, poor nutrition, pesticides, viral diseases. So just be cautious when interpreting results. If the only thing that they noticed was changes to the hypopharyngeal glands, just with a little bit of bias, interpret those results because it's not quite as devastating. We don't know the long-term effects is the other thing. So it is entirely possible that they could recover from these changes and go on to continue to be productive worker honeybees if I hadn't made them into slides. So the last part of my experiment, like I said, is we think that we have a compound that might be able to treat some of these viral diseases. And we're going to start by testing them in a cell line of honeybees. So we actually have isolated cells from honeybees. We grow them in culture, and we can use them as an an ex vivo way of testing some of these viral diseases and compounds. But thanks, Liz. I love any opportunity to talk about my topic. So thank you for bringing that up.
Okay, so there's a few questions. The one on Chalkbrood we'll save for Liz. The one, which lab can I submit bee samples for viral load analysis? That was the lab out of Maryland.
If you're in the US, yes, Beltsville, Maryland. It seems like in Canada, Beaverlodge is the place to go. So maybe Liz can talk a little bit more about that.
Yeah, the National Bee Diagnostic Center, which is currently headed by Patricia Wolf-Vega, does viral screening for a small fee. So she will do it for US beekeepers as well, but you get to figure out how to get it through customs.
Yeah, please don't mail stuff unless you talk to somebody first.
So what I'll do is I'll post those in a reply to all the participants, so you'll get a link to both those labs.
Thanks.
Let's see. There's one question around testing honey for viral loads. Is there a place we can do that?
That's an excellent question. I'm not sure if Beltsville will do honey. My understanding is that they mostly do dead bee samples. I will say that some of the viral diseases have been detected in honey. So it is a way by which viruses can be transmitted. But no risk to you personally. Like, none of these are zoonotic. By eating honey from hives that are infected, it won't hurt you or anything.
Good. Another one was about participating in the Sentinel program. I guess there's a 10 hive minimum.
Oh, cool.
Because you need more than 10 hives to be part of the program. And I guess any advice for folks who have less? Is it a citizen science project with your bee clubs where you collect money to send samples out? Because
that's such a good idea.
Because there's a lot of the shipping cost to send it to Maryland. So for the folks in the US, so there's ways of doing it. So if you have less, just collect bees in a Ziploc bag and email some of these scientists, and they're happy to help you. I've sent bees to Patricia, and I've sent bees to, that's how I met Marley, is by sending her dead bees across the border.
We fixed them in ethanol first, so it was all completely above board.
Yeah.
But yes, that's why it's so important to talk to somebody first, because there are some pretty important rules and regs to keep you on the right side of the law.
Exactly. So if you do it, then the key is to share your results, because that's the challenge with beekeepers, is we don't share a lot of our results. And the thing with citizen science, the only way it's going to work is if you have a bunch of sick bees, let people know. Please. Because if you keep it a secret, that's the challenge with science, is if a tree falls in the forest, nobody knows about it. Did the tree really fall? And you can say that about studies and sicknesses and all that type of stuff. So there's a lot of choices there. One comment regarding winter bees: could the resistance be due to juvenile hormones maintained in the winter bees?
Oh, that's an awesome question. So I don't know a ton about like hormonal physiology, so that is not my area of expertise, but the juvenile hormone is amazing, and it does so many different things, even in adults or pre-pupae. So it's a really good thought and would be worth looking into.
Can I add to that? My name is Lisa. I'm in Alaska. I asked that question. You were talking about the hypopharyngeal gland, and I understand that winter bees who maintain that juvenile hormone throughout our winter, which is very, very long and dark and cold, can actually, depressing, can transmit that juvenile hormone to other bees through the hypopharyngeal gland.
Whoa.
And so, do I know something you don't know? Oh my God.
You know a lot I don't know, I'm sure. I'm very niche.
So I'm wondering with just that with the resistance that they have, they've got the, they're fat bees, they've got the phytogenin, and then they've got the juvenile growth hormone. So I'm wondering, since those are the two big differences between winter bees and summer bees that I know of, if one of those two, if you've got any ideas about which one of those two might be the one that gives them that resistance, because they are hived up real close together for a long.
The two of them are they're antagonistic. So if you want to have high vitellogen in a bee, then you have to have low vitellogen. You have to have low juvenile hormone. When you increase the juvenile hormone, then the vitellogen levels drop. So your winter bees are going to have less juvenile hormone and more vitellogenin. And the high vitellogenin in their fat bodies gives them longevity, as opposed to the bees that are transitioning from nurse mid-age to foragers, who increase the juvenile hormone and then drop their vitellogenin levels, and makes them, we thought initially that would make them more susceptible to viruses, but some of the research showed that that didn't necessarily do that. But the longevity is from, instead of doing the transition to nurse and ramping up their hypopharyngeal glands, they, instead of putting the vitellogenin there, they put it into their fat bodies and hold off on developing their, or producing jelly until they start rearing brood the next spring. And then they transition from essentially an immortal bee that can live for over a year to a short-lived bee when they go through the transition. And the short life is not due to chronology. What it's due is to the temporal polyethism, where they shift from nursing to mid-age to foraging. And once they initiate foraging, you got each 12 days and they're going to be dead. So as long as they maintain in either the diutinous physiology or nurse physiology, they can live for a very, very long time. When they start making that shift into mid-age bee, and then especially start foraging, and that is what really limits their longevity.
I'm wondering if that immunity boost, for lack of a better term, is transmissible with the pheromones that are released with the hypopharyngeal gland.
Okay, well, it's not pheromones.
I read somewhere that they can transmit.
But there's both the hypopharyngeal gland and the mandibular glands. That's what make the jelly that is fed and transferred. And the most recent research is indicating that there's transgenerational immune priming, not only via the ovary with the vitellogenin, but transgenerational immune priming via the jelly with microRNAs, dsRNA, and who knows what else. But that makes total sense. So the jelly is like the colostrum that a mammal mother gives to their babies to expose them to the pathogens, or to the antibodies to the pathogens. And in bees, which don't have antibodies, what they do is transgenerational immune priming via the jelly, which may be why we see these waves of things like Chalkbrood and all the other plagues. That if you kept bees long enough, what you're going to see is every few years a plague of something comes through, and everybody throws up their arms. Oh God, all the bees are going to disappear, and then they all try all different things, and then it stops. Everybody goes, oh, it's because I did this or because I did that. But if you look at the control group of beekeepers who did nothing, it disappears just the same in their operation. So the best I can tell is what you have is this transgenerational immune priming, that when you get a new variant of a pathogen, whatever it is, and we saw this when the Australian bees were brought into, I don't want to step on you, Liz. Are you going to talk about the Australian bees coming in when we, during colony collapse order, and what happened with them in California with the Chalkbrood?
Nope, I'm only talking about the Canadian stuff.
Okay, so when, for example, I was in Australia shortly before the big collapse, before the beginning of CCD, which happened 4 years before Hackenberg brought it to our attention. Okay, and it happened when there was a huge shortage of bees in California for the first time, the winter of 2004, 2005. That's when CCD really hit hard. And then they opened the borders for us to import Australian packaged bees. I had just been to Australia the year before and was looking at their bees with Chalkbrood, and they have Chalkbrood endemic there. And I said, your Chalkbrood looks a little bit different than ours in California. And then now their bees were resistant pretty much to that Chalkbrood, but it was endemic out there. Every single Australian package that came to California, the queen had to be replaced, otherwise the colony would collapse from California Chalkbrood. As soon as they got exposed to California Chalkbrood, whatever variant we had as opposed to the Australian variant, their transgenerational immune priming did not suppress that California chalkbrood in the Australian bees, and every single beekeeper had to replace their queen. Otherwise, the colony would completely get overwhelmed by chalkbrood. So this is what I see long term is this: the bees they build up population wise. For one thing, genetically, by weeding out those that are susceptible. The other is also appears to be transgenerational immune priming that allows them after a couple of years to suppress it. So we see this European Fabric variants. We see this with chalkbrood in our operation, and we see it with other things. So it's a very dynamic system out there.
Good stuff. So, and I guess, Lisa, the key for us is you got to keep your bees, winter bees, as long as possible, or they'll die too early. And that's what happens to a lot of beekeepers is they take the wrapping off too early. They do all these things that kick that. I do it on purpose. I put my pollen patties in March, and first pollen was actually early June this year. So,
oh yeah, this is what happened to us this year in California. Is you screw up the spring turnover.
Oh yeah,
the spring turnover is critical, and what we got is we had a extremely unusually warm winter here in my elevation, where the bees started getting nectar and pollen in middle of December before Christmas, which is that never happens here. What it did, it took the bees out of the Titus state, and they initiated brood rearing, and they thought, great, they're going to build up really good for almonds. And then a big freezing snowstorm came in, dropped down to 20 degrees Fahrenheit, and buried all the hives in snow. It was too late for those bees. They had already transitioned from the diutinous state to the nurse bee state, had all this brood, and they had used up everything they had. And then they suddenly had no pollen coming in. Yeah. So that brood can cannibalize the brood, but they're not going to be able to rebound and restart the nursing the way that they did before. They're going to have this short longevity, and it just kneecapped all of our colonies across the board. And that's what's called an unsuccessful spring turnover due to a weather event. And it happens a lot on the East Coast. We don't see it happen much out here, but we got hit hard by it this year.
So last comment on that one, I was going to say that's where my insulation comes in. It basically, the bees can take a -40 and just float off of it. So we'll have to move on to the next speaker. Marley, thank you very, very much. It was a great presentation. And you may get some questions. So if you're okay, I'll share your email with the group.
Please, I would love that.
Okay. And yeah, you please feel free to stay on as long as you want, because I'm sure you can ask some hard questions to Liz. Return the favor.
Oh no, I don't know anything about fungal diseases.
Then it's something to learn then. So
that just means there's more room for questions.
Yeah.
Love it.
So just a quick bio on, so Dr. Liz Walsh is a newly hired research scientist with the USDA-ARS laboratory in Baton Rouge, Louisiana. She recently completed her postdoctoral fellowship with Dr. Steve Pernal of Agriculture and Agri-Food Canada at the Beaverlodge Research Farm in Alberta. Liz's postdoc was spent exploring the links between honeybee health challenges and honeybee biomarkers as part of the National BeeCSI project. But she also did work with AFB and Chalkbrood, exploring stock variation, asymptomatic versus symptomatic infections, and more. This was all very different than her dissertation work, which was done at Texas A&M University with Dr. Juliana Rangel, where Liz explored the impact of miticide exposure in immature queens. Liz is pleased to be well into her teenage years as a beekeeper, since she began keeping bees as a young high school student in her home state of Wisconsin, and is proud to serve the beekeeping industry through research initiatives. So with that, Liz, the floor is yours. And for the panelists, if you can just mute your videos, it just helps with bandwidth.
Thank you so much, Etienne. I really appreciate your introduction. So without further ado, we're gonna go into they want their mummies, variants of chalkbrood symptoms in honeybee colonies. All right, and Marley modeled an introduction slide so well that I knew I would have to do one too. So as was said in the introduction, I began beekeeping in my sophomore year of high school. So I'm what I refer to as a teenager beekeeper. So I know quite enough to be dangerous and yet think I know it all sometimes, but don't worry, the bees always put me in my place. After completing my bachelor's in Wisconsin, I went to Juliana Rangel's lab at Texas A&M, where I defended in the fall of 2019 and graduated late that year. I did work on queen health, so I got to learn a lot of ways to assess the queen, and I was particularly interested to see how the queens were impacted by early exposure to miticides, so the pesticides that we as beekeepers put into our colonies. I found that it was a very mixed bag. Sometimes the miticides had negative effects. One time they plausibly had a positive effect, and sometimes they had no effects at all. It just depended on what metric I was looking at. From there, I went to Dr. Steve Pernal's lab in Northern Alberta. So it was quite the climatic shift for me to go from Texas to Northern Alberta. So Beaverlodge is north of Edmonton, if you're familiar with that area. And it was my absolute pleasure to be part of his lab. I was hired to do work with BeeCSI, which is a project headed by Amro Zayed out of York University and Leonard Foster out of UBC. They're doing this national study in Canada with many, many labs and over 30 people. I was privileged to be one of them. And we're still examining the data. There's quite a lot. The goal is to find unique biomarkers that correspond with specific health stressors that the bees encounter, in the hopes that the National Bee Diagnostic Center could use those biomarkers to identify beekeepers' submitted samples. And we're on our way to actualizing that goal, which is pretty dang cool. Now I just moved just a handful of weeks ago to Baton Rouge, Louisiana. So now I'm on the Gulf of Mexico. Just got in on the tail end of the tallow flow. And so far, so good. So let's shift to the Chalkbrood project. So you know me already, and I hope you can see my mouse. If you can't, it's in the top left corner on my face. You know me. The next one to my right pictured is Marlene Palliard. She's in Quebec at the University of Laval and Cresade. She's a fantastic beekeeper and a pleasure to work with. This project had 2 geographic locations, one in Northern Alberta in Beaverlodge and one in Quebec near Laval. And we were able to do that because Pierre Giovanazzo, pictured here on the bottom right, so generously agreed to collaborate with us. So they were the Quebecois branch of this study. And I won't be presenting data from that location today. And then here on the bottom left, we have Dr. Steve Pernal, who I had the absolute pleasure of working with from 2020 up until just a few weeks ago. So let's jump into it. So Ascosphaera apis is a fungus that has a worldwide range. And if you've been a beekeeper long enough, you're going to see it. It causes immature bees to desiccate and dry, they die. They become what we colloquially call mummies, and they could be white mummies or black mummies. It just depends on what life stage Ascosphaera is at. And what's really interesting is even though this fungal parasite is so incredibly ubiquitous, there's no registered chemical treatment in North America. And beyond that, we give new beekeepers who are encountering it what I find to be the most infuriating advice, which is, oh, just keep strong colonies. And everybody here who's been a beekeeper long enough knows that that's easier said than done. And also the silver bullet to fix everything. You have problems with X, Y, and Z, just keep strong colonies and it'll clear right up. Unfortunately, it's not so easy to keep strong colonies. Chalkbrood is interesting to me for many reasons, but in part because it can be both symptomatic and asymptomatic in a colony. So by the time beekeepers notice it, notice these mummies, it's commonly quite late in the infection, and people will see the mummies piled up on the bottom board, like this picture that Dr. Harris took, or right outside the colony entrance. By the time it gets to this stage, it's quite a severe infection. All right, so how do we define social immunity behaviors? Marley touched on them before, to my great pleasure. And I define them as traits in which honeybees do behaviors sorry, to reduce infection frequency, infection intensity, and ultimately disease transmission. This is a relatively broad definition. So I'm going to include this wonderful figure that Mike Simone-Finstrom had in a review paper from 2017. In it, he talks about how social immunity behaviors can really all be present at varying levels, a variety of the time, but he kind of puts them on a spectrum. And he says that you can group them into constitutive mechanisms or inducible traits. And to be fair, he doesn't really say you can group them. He just says they tend to fall along this spectrum somewhere, where it's a constitutive mechanism in that the bees are always doing it all the time. It's part of their basic biology. And it has a side benefit of being a behavior which reduces infection frequency, intensity, and disease transmission. Or it's an inducible trait where the bees are faced with a health challenge and they begin this behavior in response to it. Now, the two behaviors here that I'm particularly interested in sharing information with you tonight are propolis, which bees make after collecting tree resin, and also hygienic behavior, which Marley also spoke about in regards to Varroa control. My ultimate umbrella research question was, how does honeybee stock impact social immunity behaviors after exposure to chalkbrood? So, as all scientists do, I get to tell you how I plan to answer this question before I show you the lovely, lovely data. So I had 3 different stocks. I had an Albertan stock, a Quebec stock, and a New Zealand stock. And I did this because the Canadian beekeepers that I had the absolute pleasure of working with reported something similar to what Randy said earlier about the Australian queens. The beekeepers said that the New Zealand queens were good to get broods started early because you can rear queens in New Zealand much earlier than you can rear queens in northern Alberta. But that in northern Alberta, those colonies had to be requeened as soon as possible with more locally bred queens, because otherwise the colonies would fail, commonly because of chalkbrood. For this study, although we had 3 stocks, we only had 2 distinct locations where we ran mirror experiments. And so these locations are marked with stars, Alberta and Quebec. And so we did a stock exchange where we flew queens out to each other, and then we introduced them to small nucleus colonies and waited until the population had turned over so that we knew each colony was very much a colony of a specific stock. We had also bought New Zealand queens and had established them in nucleus colonies to do the same thing. So each location had 3 stocks. Once the colonies were established, we inoculated them with Chalkbrood and I colloquially called them the plague patties. I spiked these pollen patties with Chalkbrood and then gave them to the bees. Now, let's just do a very quick stock comparison, just so that you have a very solid footing into what my thought process was going into this. So I knew some of the history for the Albertan and Quebecois stocks. I knew that the Albertan stock had previously been managed by Dr. Abdullah Ibrahim, who helped establish the hygienic behavior assay in Marley Spivak's lab. Abdullah is just a gem, and I cannot tell you what a joy it was to work with him. The stock that his breeding program at the Beaverlodge Research Farm was quite intensive, but it did lapse about 8 years ago, 6 to 8 years ago, depending on how strict your definition of lapsed is. So it was intensely managed, and then it was not. The Quebecois stock has actively been bred for hygienic behavior primarily, but for several other things too, for the past 11 years. They have a very, very strict and regimented breeding program, and it's very interesting to hear talks about it or to read about it. So I would highly encourage you to check it out if you get the chance. The third stock was the New Zealand stock. It was not bred for hygienic behavior. And unfortunately, for the Canadian beekeepers, at least, we know that the bees very rarely encountered, compared to the Alberta rates, rarely encountered Chalkbrood at all. And when they did, it would have been a different strain of Chalkbrood that they encountered. This is in quite stark contrast to the Quebec stock especially, which very commonly encountered Chalkbrood of the Canadian strain, as well as, well, presumably of the Canadian strain. I haven't personally tested that, but I feel safe saying that. And also the bees from there in Alberta who moderately encountered Chalkbrood as well. So here's my caveat. I will be discussing the bee stock data from the Albertan and Quebec stocks at the Alberta location. And that is all I'll be talking about today. Part of that is because, as the beekeepers suggested, our New Zealand stock had extreme attrition before we could even get the inoculation patties into them. We had lost, depending on the year, approximately half of our colonies. And quite a lot of those were due to Chalkbrood. I called them my overachievers who got Chalkbrood before the experiment started. But sometimes the queens seemingly failed or were spontaneously superseded. So I really don't know what happened to all of them, which gives room for future work, I guess. So now that we have the groundwork established for what I was working with, how was I working with it? Well, I did propolis envelope collections and measurement. So here you can see a as opposed to some of the methods that Mike Simone-Finstrom still uses of roughing up the inside of the box, I used the methods that he used at Minnesota, as well as the ones that Hinata Borba used at Minnesota, where I cut propolis traps up and I screwed them in to the sides of brood boxes, which allowed me to collect the propolis quite easily. I also did hygienic behavior assays, which we could have an hour-long seminar about how to do them, but suffice to say that Abdullah and I did them using 2 PVC pipes that ultimately had the correct area of brood, and that we then flash froze with liquid nitrogen to be assessed 24 hours afterwards for removal or lack thereof. I also measured the number of mummies that the bee colonies were removing from the frames per day. And I did this via dead bee traps in the front of the colonies. They have these charming little drawers here. So the bees, the undertakers, chuck the mummies right out the front step and it ends up in this tray. I also had plastic, corrugated plastic that I had in the bottom boards of each colony. So I would pull that out and examine the bottom boards as well. And I added those totals to get the daily mummy count. All right, so data, so much data, guys. So much data, I still am not quite sure how to process all of it. So thank you for your patience as you help me figure out what matters. I will note now that I tried to denote what year data was from in the banner. The data I'm presenting tonight is from 2020 and 2021. The 2022 data has just finished being collected. And so this is the data you'll hear about. So in terms of propolis envelopes, there was no statistically significant difference between the Quebec stock and the Albertan stock, which, okay. However, when we split those stocks into inoculated or uninoculated groups, there's no statistical difference either. And so remember that bit about the intro where I talked about a constitutive mechanism or an inducible response. This data here, where the inoculated and uninoculated groups are broken down, shows that this, in this year, in this study, was likely not an inducible response, because otherwise we would expect to see a difference in the propolis envelope mass in the inoculated versus the inoculated groups. And this is what I just said. I have these lovely little signs up just in case I get nervous, because for some reason I get very nervous sharing these talks virtually, but not as much in person. So if I go to heck in a handbasket, you've still got the sign. All right, so here is the propolis data from the following year. And again, I have it broken down by stock here, and there's no statistically significant difference. Again, there's also no difference in inoculated versus uninoculated stock, which is interesting, given that there's a slightly different pattern this year than the previous year shown. So take a look at the pattern here, where the Alberta bees actually had larger propolis envelopes than the Quebec inoculated bees, and vice versa with the uninoculated bee stocks. And compare that to this, where the Quebec bees consistently had slightly larger propolis envelopes. Again, these differences were not statistically significant, but it makes me wonder if the sample sizes had been extremely larger, if we would have seen this consistently or not. All right. So again, there was yearly variation also. So I didn't explicitly say that, but the Albertan stock had larger propolis envelopes in the infected group in the second year. So things were not super, super consistent. Here we get to the first significant data, where we see that the Quebec stock was significantly more hygienic than the Albertan stock, which makes sense, right? Because the Quebec bees are in an active breeding program that, among other things, uses hygienic behavior as a breeding marker. And the Albertan stock had that, but it has lapsed. This pattern does hold true if the colonies are broken down in inoculated and uninoculated groups. However, these differences were not statistically significant. So again, if we think about that constitutive mechanism versus inducible behavior, we're seeing with these results that hygienic behavior is a constitutive mechanism in this case. The bees are always doing it, and they're doing it regardless of disease status. And they're always doing it, but to varying degrees, as you can see. All right, so don't freak out. But I wanted to show you a little bit about the mummies. So this is only 1 year's worth because I'm still trying to figure out how I should best process this. And so here we see these tiny lines per day showing the mummy production in a given treatment group and stock per day. So we have the Alberta inoculated bees, the Quebec inoculated bees, the Alberta uninoculated bees, and the Quebec uninoculated bees. It's very difficult to see, and I recognize that. I just wanted to give you an idea so you could see that the red and brown colors are consistently the ones that are shown to fairly large degrees sometimes. So that's showing that our inoculations did work. And that our uninoculated bees, although they occasionally will show a mummy or two produced, they really don't have much at all. That's also a good reminder to say that because Chalkbrood has historically been thought of as a parasite that's very affected by microclimates. So for instance, if you have a yard in a low area that's swampy or very humid, traditionally those are the yards that are most thought of to be most at risk for Chalkbrood. But there's not a lot of compelling data to show how infectious it is within a given yard. Here we're seeing that there are a couple of control colonies that were uninoculated that did produce a small number of mummies, but it was very, very small. And here, if you look at the number of mummies produced over the entire summer, this is incredibly small compared to over 300, for instance, or over 100, for instance. Here, these asterisks denote statistical significance. And so we see that, as we would expect and hope, there is a statistically significant difference in the inoculated and uninoculated groups. Makes sense. But there's also a statistically significant difference between the stocks within each treatment group. And interestingly enough, again, we have this inconsistent pattern where the inoculated Albertan colonies produced far, far more mummies throughout the summer than the inoculated Quebec colonies. But in the uninoculated groups, that's the opposite. So that's very interesting to me because I don't know why that is, especially because they were all in the same apiary site. They shared pallets with each other. So it's exciting because it's more to find out. So you'll also get the dubious honor of getting this presented to you first. So because of that, bear with me because it looks like my animations are a little off. In addition to all of the methodology that we've talked about and the data that's gone with it, I also had weekly frame inspections where we went into each colony, we examined each side of each frame, and we counted each mummy in each frame. I also did weekly collections of nurse bees. Here you can see the ones that are provisioning the larvae are nurses. And then I also did weekly collections of fifth instar larvae. And the reason I did that is because old larvae are the ones that, as far as we know, are the most susceptible to Ascosphaera, to Chalkbrood. And so I reasoned that if there was any possibility of discovering what the Ascosphaera threshold per individual larva was, we would have to collect larvae. So we collected 5 seemingly healthy fifth instar larvae per week, and then later on did PCR screenings to see if they tested positive for Chalkbrood, even though they were seemingly healthy.
Liz, could I ask you, those 2 photos, were they taken during your collection?
No.
Okay.
No. I nabbed them.
Something distinct about those photos that I would question then.
Yeah, no,
okay.
I nabbed them from the internet. They're anonymous. They're
both very hungry colonies.
They are. They definitely are. There's very little royal jelly. Yeah. All right. So this, so here, statistical significance is denoted because I compared the colonies in 2020 to the colonies in 2021, the adults in 2020 to the adults in 2021, and so on and so forth for the larvae. I did not compare the colony adults and larvae within a year together because I'm measuring different things and it doesn't seem fair, guys. So here on the y-axis, we have percent Chalkbrood positive in the colony, the adults, and the larvae per year here on the x-axis. What this is, what I'm trying to communicate here, and you can tell me if I'm doing it effectively or not, is that in 2020, 60-some% of the colonies at some point, at some sampling day, regardless of what, every single day there was some sampling event. At some point in 2020, 62 or so % of the colonies had a mummy. Some of them had a lot more than one mummy. The frequency is not depicted here in any way. This is just the % of colonies that ever had anything to do with Chalkbrood. Here we see the results from the pooled adult samples. So remember, I collected nurse bees. About 86 or so % of all of those, and they were pooled per colony per sampling day. 86 or so % of them tested positive. And the larvae—
Did you tell us what the test was on the adult bees, on nurse bees?
PCR. I did
okay.
PCR screening.
Thank you.
Yep. And so I did the same with the larvae, the pooled larvae, groups of 5, seemingly healthy fifth instar larvae per sampling day per colony. You can see in 2020, there's clearly way more positivity that was asymptomatic that we never would have known about than there were symptomatic infections. This all changed in 2021. And so there were a few things that were different about this. So one is that the Quebec bees were very resistant to having any Chalkbrood symptoms in Quebec, far more than they were in Alberta. So in 2020, we doubled the dose. And so we got a lot of colonies. Here you see we had about 92% of all the colonies in the experiment had at least a mummy at one sampling time point throughout the summer. That is a ton. However, don't forget, there were very, very few in the uninoculated groups. These are 1 or 2 mummies per colony in the entire time we examine them, as opposed to many more colonies in the infected ones. So, however, and possibly because of the heat dome, the bees were able to fight this infection off very quickly. So much so that the only real positive adult and larva samples were early in the summer. They were able to kick it in 2021, but they weren't able to do that so much in 2020. And my current hypothesis is that that's likely due to the extreme weather difference. These colonies were at the same apiary location both years. They were the same stocks, grafted from the same groups of queens in both locations. So I don't know how else to explain that, but it's very fascinating to me. Oh, and here's
Are you saying that it was warmer in 2021?
I'm saying Alberta had a heat dome that was terrible, Randy.
Okay, that's exactly what I would expect the effect would be.
Yeah, so we're nearing the end, I promise, and then we can chat to our heart's content. So what are some of the takeaways here? So we can see that the Quebec stock is generally more hygienic than the Albertan stock at the Alberta location. Although I didn't show it, their stock was way more hygienic than the Albertan stock at the Quebec location, which is really interesting to me because it says something about how even within one country, the environments are different enough that the bees take notice. Unfortunately, there weren't very exciting patterns in hygienic behavior or propolis envelope mass in regard to Chalkbrood state, which was kind of disappointing to me because that was the part I was really excited about initially. But it's still an interesting story to me because I really would have expected there to be very clear patterns between those two social immunity behaviors and state. So interesting. But what it shows us is that these two mechanisms are very much on the constitutive end of the spectrum rather than the inducible trait end, at least for this experiment and for these bees. Currently, we're doing qPCR on the individual larvae and adults that we collected. I really am very taken by the idea that we could figure out a threshold where an asymptomatic infection becomes a symptomatic infection. And it strikes me that that would be very important to move forward with. I'm also still crunching the data. And so now
Liz, just a quick question first. Difference between PCR and qPCR, just for the audience.
Good question, Etienne. So PCR is a polymerase chain reaction. It's just a way to molecularly determine if a particular gene of interest is present or not. So in my case, there are very well-developed methods where a particular gene that is specific to Chalkbrood is screened for, and you find out if it is there or if it is not there. That's the data that I showed you. Then there's qPCR, quantitative polymerase chain reactions. And these are where you can quantify the number of that gene that you're interested in. And so by doing that, you can get an idea how many copies of the gene is there, what degree was this larva infected or parasitized, as opposed to this other larva. So you can find out, you can say, well, this larva had less than 10 copies of a Chalkbrood gene, whereas this other larva had 500, as an example. So how does all of this apply to you? The information that varied in the locations between the hygienic behavior in Quebec and Albertan stock was super interesting to me. And I'm sorry I didn't show it. I really wanted to make sure there was enough time for a robust discussion. But even in the data that I showed you, you can see that the stocks are very much suited to the environments that they're reared in. The New Zealand stock sadly was not able to cope with the conditions that it encountered in northern Alberta. And similarly, the Quebec stock didn't perform as well in northern Alberta as it does in Quebec. We also have, and this is, I'm not gonna get preachy, but if I was gonna stand on a soapbox, it would be this one. So we need to keep in mind as queen rearers that are hopefully queen breeders, not just rearers, that different traits need their own breeding assessments. I would have bet so much money that hygienic behavior and propolis envelopes would correlate, and that furthermore, they would be incredibly significant in terms of activated, induced or not by disease state. They were not. We need to keep in mind as bee breeders that different traits need their own breeding assessments, and that throughout the entire process, we need to constantly be selecting the best mother colonies. Even though the Alberta stock had stringent breeding requirements, it lapsed for just a bit comparatively, and it's already losing the traits that it was bred for. So we need to keep these things in mind. And furthermore, we should also have this idea of actual assessments of success versus proxy measurements. So an actual measurement of success, for instance, in this talk would be, well, if I'm breeding the most Chalkbrood resistant bee that I can, I'm breeding from colonies that had 0 Chalkbrood always, not a single mummy to be found ever. A proxy measurement would be hygienic behavior for this particular work. Also, and I love this, we had one colony that I had to eject from the experiment because it had 2 laying queens in it. And because we were going in anyway, we monitored it. We found both queens for about a month and just over a month. They're both laying. They were seemingly in harmony. But obviously I can't compare that colony because who knows what the parentage of the different workers is. But it was a good reminder to me that although I am in my teenage years as a beekeeper, the bees don't read the same books that I read. So I believe this is the last slide before the acknowledgements. And these are my new coworkers. Frank Rinkovich, Mike Simone Finstrom, Kate Eiley, Ariane Avalos, and Vincent Risoglio. It's my absolute pleasure to be working with them. Frank and Mike and I are doing a Chalkbrood experiment that's starting next year, which I'm very, very excited about. There will be stocks involved in this as well. Kate and I are also doing a stock experiment looking at pupil signaling for drone removal, which I'm excited about all of this, guys. I'm just so jazzed. Ariane and I are going to be doing a drone experiment as well as an aggression study, which I'm quite excited about because I think that drones are so very understudied. And because they are half of the breeding equation, we really need to up our drone game if we're gonna have strong breeding programs. And I say that on a continent-wide basis. Vincent and I are getting ready to do, well, not getting ready to, but in long term, we hope to do a nutrition study, possibly with pesticides as well. So it's my pleasure and privilege to work with these fabulous people. I've been so lucky to go from wonderful lab to wonderful lab. I can't wait to share the next stuff with you. And And this work would not have been possible without a large variety of funding sources shown here quickly. And also, and to me, more importantly, I really need to give my heartfelt thanks and appreciation to Dr. Abdullah Ibrahim and Dr. Rasul Bahraini. Both of them helped me to varying degrees in this project. Courtney McInnes is a grad student of Dr. Pernal's, and she's just been wonderful to bounce ideas off of. Vivian, Kellen, Diana, and Timothy were all undergraduate students that helped in some capacity or other with the experiment in 2021. And Shelley Huang, Eva Marie Smith, and Natalia Claremont were the undergraduates of the lab this year. Shelley Huang was so incredible. She took the reins when I left halfway through the experiment, guys. It's not every undergrad you can trust to do something like that. So I can't say enough good things. Here's a picture of all of them in the chaos of getting the experiment ready into the colony's special propolis trap boxes with their dead bee traps at the ready. So lastly, absolutely don't hesitate to email me if you have any questions that we aren't able to get to tonight. It's been my pleasure to share with you, and I really appreciate this opportunity. Thank you.
All right, can I hit you first?
Sure.
Have at it.
Okay, wonderful research, Liz. And what I really like is your response to when you were surprised by the results that your hypothesis is not supported, because that's what happens with me in field research almost 100% of the time. And I've just explained to my two new assistants today, that is the exciting thing, because if it's contrary to what your hypothesis was, you have a chance of learning something totally new and maybe unexpected and serendipity. And I gave them some examples of research before. So anyway, I'm glad to see you with that attitude. Okay, two questions. One, years ago, I read that chalkbrood also can be found in bee bread, but then Kirk Anderson's research showing the high acidity of that. Did you check the bee bread?
So we did, but right after I made it.
No, I'm talking about the natural.
I'm sorry,
I
misspoke. Yes, no, we did not check the natural bee bread.
Okay,
we only checked the pollen patties.
When you did your PCR, did you include the guts when the homogenate?
It was only the guts. We dissected the guts only.
Okay, so if it is found in the bee bread, your PCR could have shown it was only in the bee bread and not infecting the bees. That could have been, that's a possibility that you could have been picking up.
That's why, yes, you're correct. That's partly
why. So I would
why
suggest that you do an analysis on the natural bee bread to see if that may be the alternative host for the chalkbrood, the reservoir of the inside the colony.
It would be cool. Yeah.
Okay. The second question is, okay, I'm a quick guy. Okay, go ahead.
I have one clarification because I didn't say so. Don't tell Steve. I neglected to mention that for the adult nurse bees that we sampled, we dissected their guts and pooled an aliquot of their guts for PCR. We did not have the entire bee that we worked with.
Right, so since the nurse bees contain pollen in their guts, then that would be, you got to make sure that you weren't picking up a contamination from the pollen independent of actually an infected bee.
Yep.
Okay.
It was really neat.
Second question on your temperature thing. Okay, so chalkbrood apparently can only grow in a bee that has a temperature low enough that you have a high enough oxygen content. As the temperature in the body increases slightly and the oxygen content drops, chalkbrood, from what I've read, is unable to reproduce. Do you have any thermocouples in your hives of the brood to see if the temperature of the brood was different during that heat wave?
So we did not. I would have loved to, but 60 of those remote thermometers was too big of a budget.
Oh, yeah, even in a couple of colonies, just to see if there was a difference on whether it's not the dampness of the low areas, maybe that little tiny bit of difference in temperature might be, because chalkbrood generally for us disappears when it warms up, and that may not be the variable. But
oh, yeah.
Yeah.
It
The
might
problem with
correlate.
so much of the stuff in beekeeping is that you say, oh, well, something disappears during the summer. Well, it's not just the date that's the variable. There's a whole lot of variables associated with the summer or with the winter, and teasing out those variables is often difficult.
Yes.
Any comments on Randy's comments, Liz, before we move on to MedHat?
I think we're good to go.
Okay, good.
I will say too, it was exciting. This is just a comment you've inspired. I was really, really tickled to get to work on something so different than queen health and pesticide health. And part of that was because I got all these new techniques that are very standard in many kinds of biology, but that I hadn't had the opportunity to practice yet. And so it was cool to learn about aerobic versus anaerobic growing conditions of media and all sorts of things. I still get quite a kick out of it.
I'm excited for you and looking forward to the rest of your research over the rest of your career.
Thank you.
The floor is yours.
Yeah, congratulations, Liz, and you are moving into a wonderful lab. I have a daughter lives in Baton Rouge, so whenever I visit, I always visit the lab out there. Nice. So that will be a good opportunity to meet you. Yeah, excellent research, and one of the things about the propolis envelope, have you considered the situation in Deschambault where Pierre Jovenet's does work, lots of trees and lots of sources of propolis. However, in Alberta, in the Peace River, I lived there 20 years, so I still live here in Alberta. There's nothing there. And I noticed from my experience that we don't have much of propolis out there in this part of the world. So that's might be considered like in your results to see the difference about like, there's no sources of propolis. So
MedHat, you're trying to start a fight.
No, I'm not.
No, you're good. I agree that there's a lot less propolis in northern Alberta than I was used to seeing, but we did choose an apiary location in part because of its close proximity to a very robust wooded area. So there was propolis. Yep. It wasn't as much as I expected though. You're right.
The second thing, just also knowing, yes, I used to get these calls about the imported stock and stuff like that. Mm-hmm. With the combs, established your colonies, was they brand new?
They were irradiated. So they were not brand new, but they were irradiated.
Okay. Okay. Yeah, that's a good thing. If I may ask, how many gigs? Because apparently Chalkbrood requires higher gigs more than the AFB.
I'm sorry.
Higher gigs, like higher power of irradiation, more than the spores for AFB.
Yeah, that's a good question. I would have to double check that. Yeah. Yeah. We did use new equipment for everything else, but the frames, we did use drawn out frames that were irradiated, and I'm afraid I don't know exactly.
Yeah, I'm asking that because we tried actually with the facility. I'm sure you are using the one in BC, and they are set up for American foul brood, but with the AFB, with the Chalkbrood, it needed higher power, and that's the time we tried to negotiate with them how to increase it to make sure everything can be done, because as you know, they buy the bees from overseas and put it in infected colonies. Not everybody irradiates, and reinfection all the time going. So the problem becoming, is it coming from old infection and the transfer from the comb, or from the new bees coming in?
Well, so and that's well, and I was very, very worried about that. And that was part of my my reasoning for showing that tiny graph, to be like, look, see, the uninoculated groups are mostly like, they're mostly good guys. So yeah, you're definitely, you've got good.
Yeah, I never really experienced any kind of Chalkbrood would kill colonies until I moved to Alberta, and I went to 1 yard, and definitely 90% of the colonies were completely dead from Chalkbrood. But nowadays, I think they continue to get the bees, and they learn how to manage it in the, by replacing the queens and taking care of that. So yeah, it's quite interesting research. Thank you.
Thank you.
I guess in the Q&A, there was a question from our friend from New Zealand around Chalkbrood in Australia and how there's a new product, green banana supplements. Have you, I guess I've heard about recently about putting banana peels and bananas in the colonies for Chalkbrood, but is it something you looked at or heard of?
I have not looked at it, and I had not heard about it. So all I can think of is those terpenes, those alarm pheromones. So I'd be really interested in seeing what happens.
Okay, maybe it's hot bees.
Some years ago, I was working with a mycologist, and he was culturing Chalkbrood in petri dishes. Yeah. And I said, hey, do me a favor. Can you put some banana peel and some banana fruit into the petri dishes and see if there's any inhibitory effect on the Chalkbrood? Because that's a very common cure worldwide, to put bananas in a colony. If you study Chalkbrood, you're going to come across this all the time. Okay, and he's found no inhibitory effect from the banana peels. Now, that doesn't discount that maybe the amyl acetate in bananas, which is now is the same alarm pheromone, might induce a behavioral response or something. But as far as a straight chemical response on the Chalkbrood fungus in a petri dish, he did not find any effect.
Yeah, I always love thinking about those things where it's like, okay, was it toxicological? Was it some sort of physiological biochemistry stuff, or was it a behavior that was triggered?
Mm-hmm. I guess I have a nook that has some Chalkbrood right now that I may just see what happens.
Banana it up, man.
And I've got some spotty bananas. Okay, so any other questions out there? Comments?
Yeah, you know what? You're talking about the freeze-killed hygiene. And when you say that the test, it should be always specified. This is freeze-killed hygiene test. I was showing, I'm training two new assistants today, and I said, hey, take a look at these colonies. So this is uncapping behavior, okay? Then I said, this is varroa-sensitive hygiene. And they said, oh, you mean hygienic behavior? I said, no, varroa-sensitive hygiene. We'll see if we can find hygienic behavior. And finally, we came across a colony then in full parasitic mite syndrome collapse. And I said, now this is hygienic behavior. So the difference is between the uncapping behavior, the varroa-sensitive hygiene, hygienic behavior, and freeze-killed hygiene, which are all different things that may not directly what's her name over there in the East Coast with the new sick brood pheromone? Yeah, Kara. Anyway, so what I was showing is look at the color of the pupae being removed. If they have melanization or discoloring, that's hygienic behavior. If they're pure white, that's varroa-sensitive hygiene. If they're simply taking the capping off and the eyes are not removed, then that's simply uncapping behavior. Yeah. Three very different ones, of which freeze-killed hygiene may not have anything to do with any of them.
You should chat with Abdullah because he was very.
I know his research from way back then.
He was very. He schooled me initially because I was not being picky enough about choosing purple-eyed brood.
Okay, good.
I guess I have a question around stressors or triggers. Mm-hmm. Any idea about what makes, I guess, to go from symptomatic to asymptomatic to symptomatic? So if there's a background level of Chalkbrood, what out there what stories do you have that you've seen triggers?
So I think that environment, yes, it very much plays a role. There was 1 year where we had, so it should be noted, all of the stocks in both years, mummy production peaked on day 9. And then the beast started resolving itself. But some stocks had second blips and some did not. And I can't remember which year it must have been 2020 because we had no rain last year. But one year there was a good 2 or 3 days of straight, very hard rainfall. And that, in my opinion, caused that second peak to come sooner than it would have had that rain not happened. Because the second year, or the other year, there was a second peak in some of the stocks, but it was later than it was the year with the rain. So environment absolutely plays a role, but I also think that chalkbrood is just a lot more opportunistic than I have ever given it credit for. Before this experiment, I hadn't really been as cognizant about how long Ascosphaera can stay viable and in the environment, whether the environment is the pallets that you're using or the ground under those pallets or your brood boxes. We've had documented evidence that Ascosphaera has remained viable after 15 years of indoor and outdoor conditions during that time. So holy man, that's a long time. And it really does seem to lie in wait for any sort of stressor, as far as I can tell. And that may be a dearth, like what we're seeing now here after the tallow flow, or that may be something else. So for instance, we had, like so many Albertan beekeepers, we had an EFB outbreak in our apiary in 2020. And the colonies that had EFB were immediately ejected from the experiment and from the yard altogether. But I'm telling you, I've never seen colonies that were producing as many mummies as those EFB co-infected colonies. I think that anything that compromises the bee's immune system, which Marley talked a little bit about in her talk, Ascosphaera is ready. So.
Just a quick comment. So I ran some control hive this winter with just honey frames.
Yeah.
And we got down to minus 48 here for a couple of days. Over about 3 weeks, it was the coldest. The warmest was the minus 35. And this was a control, so no bees inside. And the coldest it got in that box was minus 20.
Yeah.
Because I had temperature sensors above and below the frame. So this was just a single. And it just, and you can actually see the lag, but the lag is so slow. So a lot of folks, and this is extreme temperatures, and people say, oh, no, Nosema will get killed by the cold if there's a dead and stuff like that. The boxes actually don't freeze.
Yeah.
Even if it doesn't freeze up here, it won't freeze down there.
Yeah, it's a marvelous design. Yes.
Oh, it's incredible. And I guess the other one for me is I've only seen Chalkbrood in transported units. So I got some nooks from Vancouver a couple of weeks ago, full of Chalkbrood. And I helped a beekeeper do her first split. And then she gave me the old queen with a couple of frames. I brought it over to my yard, Chalkbrood. Yeah. When I inspected it in her yard, no problem. And within 2 days, there was Chalkbrood when it reached my yard.
Yeah, and see, it's those stories too that really make me even more convinced that we're onto something with this idea of there's a stress, your immune system's weakened. Chalkbrood is so opportunistic, it's going for it. And moving like that is definitely a stressor. So I'm sorry to hear that, though.
No worries. They usually work through it, and it is what it is. But Madat, your hand's up.
I'm sorry. I should have removed my hand.
Okay, no problem.
Yeah. There's a quick thing.
Go ahead.
If you're looking for some way to encourage the bees to clean up the mummies, because we did a lot of work with using formic acid for varroa. but actually with the small pads was 40 mils, 65%. If you add one pad at a time, couple of times, that even though it's not really effective against Varroa, but mainly it triggers the bees to do hygienic behavior and remove the mummies out. We noticed that. The second one, some of essential oils, it can help also. Anything that triggers the hygienic behavior, that will help to clear up the colony faster from the mummies, and the colony will recover in shorter time.
Yeah,
I should write note about
that's
it.
interesting, because
Yeah.
out in the field today, where a beekeeper sent me a Thymol gel that he was saying, hey, this will take the place of Apivar, and I thought, or Apiguard, much cheaper. And we put it on 30 colonies, and after checking their mite levels, went back today, 4 days later, to see what was happening, see if there was adverse effects. And you couldn't tell any adverse effects out in front, except for a few. I saw, when we looked carefully, the occasional freshly dead bee, and one newly emerged bee pulled out. Then we went inside, and the center 4 frames, all the open brood and eggs had been removed, and the queens had moved over to the side. And so you had to get 2 frames off of center either direction to find any young larvae at all. So that irritation of, as you say, either formic acid or the thymol going into the hive, the bees just clean everything out of the open brood. They leave the pupae typically, and then the brood comes back, especially after formic. You probably noticed that after a formic treatment, the brood always comes back looking better than it was before it started. And I've always wondered, is that simply due to increasing the hygiene? Is it actually a sterilization or disinfection effect of the formic? And that still needs to be shown.
We noticed that thing, but it happens when you have really higher concentration and overpowering the hive, and that's the time you can see even the bees gets out of the hive at that time, and they are cleaning eggs, young larvae, and lots of things at the time. And you're right, once the colony recovers, the queen will lay eggs, and you find solid brood after that.
Right.
Why? Yeah. But
there's one for you, Liz.
I think too that this kind of goes back to some of those distinctions between hygienic behavior and hygiene behaviors for specific reasons too, because it seems like colonies that do exhibit high hygienic behavior, if they get on a tear, they'll take out everything and they'll just go overboard. It's like they're on a mission. And I've always kind of privately wondered if the bounce back is due to there being less competition for nurse attention and feeding rates being increased. The quality of care and nutrition may be better, but it could be several things as well.
Yeah. On your subject of exposure to these pathogens, what people often don't realize is how quickly the floral exposure is. When you get foragers flying out, and there's been studies showing this, how rapid the transmission of nosema and viruses, and I'm sure chalkbrood spores too. They don't realize that every colony, every bee in every colony is carrying viruses. Everyone is carrying all these pathogens, just like we humans are all the time. So it's the difference between carrying a pathogen, which PCR may pick up, and disease is all about failure of adequate immune response to the pathogens that are always there in the yard. And you talk about colony to colony transmission. Well, you're going to have transmission via flowers every time those bees fly, the colony to colony transmission. And then as Marley said, the drift of nurse bees, and I should say newly emerged and nurse bees, the first few days of life, their first defecation flight can just be insane.
It did not escape my notice that I don't know if it's the majority or not, actually. I would have to go and check, but enough that I noticed it and remarked on it to my undergraduate student. The majority of the control uninoculated colonies that had those onesie, twosie mummies were right away, right at the beginning. And I attributed that to drift.
Yeah.
Great conversation.
Thank you so much.
You should see us when we're offline.
Oh, so you know each other?
Well, no, in general, you know, we're all very polite when we're online.
Oh,
we're
yeah,
much
yeah.
more challenging when we're offline.
I do have to say the bathroom wait times have been pristine at this venue. I don't
Yes.
know how you manage that. I've never had such a short wait bathroom time at a beekeeper meeting.
Yes.
So with that, we'll call it a night.
Thank you, everybody.
I'd like to thank Marley and Liz for participating. It's been great to have you. And hopefully and I guess, sorry, the next talk is, I just posted it and most of you should have got the email.
Mm-hmm.
So
Yep.
we've got actually Dr. Juliana Rangel,
Okay.
maybe
You know
one of
her.
her students. Yeah. And then the second talk will be, Garrett Slater? It'll be on queens and drones. He's doing research on drones, and Juliana's lab is working on queens. So basically, that's our next talk. It'll be July 18th. And Liz and Marley, if you're interested in participating, just let me know and I'll send you the link. That's the least I can do for you. And so yeah, I'll put out email. I'll put an email out with the contact info and stuff like that.
Bye. Thanks, everybody.
Thank you.
Right.